Hi there, it's Mrs. Vance again, and we're now ready to go on to Part 4 of Unit 10 notes. This one's about variations or exceptions to Mendel's laws. So, as you might expect, since Mendel was working back in 1860s and didn't really know anything about chromosomes or DNA or meiosis or things like that, he was observing some relatively simple things, but everything doesn't always work out exactly the way the pea plants worked out. And he found that out by trying to do some work with some different kinds of plants that didn't work out quite as evenly. So we're going to figure out what that is. Okay. So in his pea crosses, the crosses always looked like one of the parental varieties. Okay. That's called complete dominance. For some traits, there are hybrids that kind of fall in between the phenotypes of the two parents. This is called incomplete dominance. In this case, neither allele is dominant over the other, and so you get some combination of the expression of both alleles in the offspring. So let's look at some examples. These flowers are called snapdragons, okay? And snapdragons come in red or white. These particular true breeding ones. So we have true breeding red ones and true breeding white ones. If they self-pollinate, the red ones always produce red offspring and the white ones always produce white. But if you cross them, okay, remember, if it's homozygous dominant, there's only one kind of gamete that can be produced. That's the dominant allele. And the same thing with the recessive one. You can only produce one kind. When these are crossed, what you find is the hybrids are pink. They inherited a dominant allele from the red and a recessive allele, was supposedly recessive from the white. But instead of being completely dominant, the red isn't really dominant over the white. They just mix together. There's a blending together of the two traits, like if you took red paint and white paint and mixed them and ended up with pink paint. That would be an example of incomplete dominance. Okay. So sometimes they're represented like this with a capital and a lowercase. Other times they're represented with two different capital letters showing that neither one is really dominant over the other. And so you'll see various ways of representing it in different kinds of worksheets. In fact, in some of the worksheets that you will do on these particular differences, you may see some different ways that they're represented. Okay? There are also genes that have more than two alleles in the population. Now, you can only have two different alleles of a particular gene, but oftentimes there are multiple ones that exist in the bigger population. A really good example of this is the human blood types A, B, and O. Now, we're not talking about the positive and negative. That's the Rh factor, and that's a totally separate blood type that has simple dominance, okay? But the ABO blood groups involve three alleles for the same gene, okay? There are four different blood groups. There's type A, type B, type AB, and type O. And all four of those result from combinations of those three different alleles. The A and B alleles are both expressed when a person has both of those, and this is called codominance. So both of them are expressed. They both produce a particular kind of receptor on the surface of the blood cells. Not a receptor, but a polysaccharide on the surface of the blood cells, and that's since neither one is dominant over the other, they're called codominant. Okay, in codominance, neither one's dominant, and both of them are expressed. There are varying examples of that in nature. AB blood type is an example of codominance. Okay. There's also various kinds of animal colorations that are kind of like that. For instance, there's a type of horse called a blue roan horse that looks kind of like a gray kind of color or silvery kind of color. When you get up close to it and look at it, the hairs on the horse are not really gray. They're actually black hairs and white hairs, and the appearance of it altogether gives it that kind of gray or silvery kind of appearance. Okay, so here we have the blood groups. Okay. If you have the A phenotype, type A blood, okay, you can have either a homozygous dominant or heterozygous with the O. O is recessive to both A and B, okay, and that means that you're producing this carbohydrate on the surface of your blood cells. Now, I don't think it really looks like a triangle. This is just a way to represent the carbohydrate that is found on the surface of the blood cells as a result of type A. If you have type B blood, then you can either be homozygous or heterozygous. Then you're going to produce the B carbohydrate on your blood cells. If you have type AB, that means you have both of these dominant alleles, okay, the codominant alleles. This is the only genotype that can produce this phenotype, and you're going to have both types of carbohydrates on the surface of your blood cells. Type O is recessive to both of those, and so type O is the lowercase, and that one has neither of those carbohydrates on the surface. Now, there are times that we will use other letters to represent the trait, and then use the phenotype initials, like we see here, as the superscript on that. You'll see various alleles represented that way especially when there are multiple ones, like this particular one with the ABO blood groups. Okay, you can write it when you're doing problems. You can just write A, B, and O. That as long as you remember that O is going to be for the same trait as A and B, as far as the carbohydrate on the surface of the blood cells. The other thing about this is also brings into something called it's also about blood types. Okay, and so when we're talking about blood types, let's look at what happens there. And we'll talk a little bit more about this when we talk about immunity. But basically, your phenotype is going to produce, you're going to have antibodies in your blood against the opposite phenotype. Okay, and so and that's going to cause problems for you. So this is how you determine what's safe to transfuse into another person. When you have a blood transfusion, the hospital or wherever you get the blood from is going to be very, very careful to carefully type and cross-match your blood with the blood you're going to receive because of potential difficulties like this. Okay, so if you are blood type A, then you have antibodies against the B phenotype in your blood. So if you mix your blood with type O blood, there's not a problem because O doesn't have any antibodies against or you don't have any, there's no antibodies and no antigens for the O to cause problems with. If you have type A blood, then you have the same kind of antibodies and there's nothing there to stir up the antibodies there. But type B blood, since the B carbohydrate is present on those blood cells, then the anti-B antibodies are going to cause the blood to clump or coagulate or clot. And the same thing with type AB, because again they've got that B carbohydrate on the blood cells as well as the A1, and so the anti-B antibodies are going to fight against those particular blood cells. If you have type B blood, you have anti-A antibodies, and so you can take blood from type O and blood from type B, but A and AB are both going to cause problems for you. If you have type AB, then you have both of those carbohydrates, but neither one of the antibodies. And so type AB blood can take blood from their blood can mix with any other blood type without a problem. And type O blood, since it has neither of those carbohydrates, it has antibodies against both of them, so type O blood can only take type O. Type O blood is often called the universal donor because any other blood type can take blood from a type O person. And type AB is called the universal recipient because the type AB person can take blood from anyone. That, again, I will reiterate, that does not generally happen except in an extreme emergency. But that's why blood banks oftentimes like to have lots of type O blood on hand, because in an emergency, when someone is losing a lot of blood and you need to replace blood quickly, then you can use type O blood as long as it matches some of the other blood types that are present, like the Rh factor. We'll talk more about blood types and antibody reactions when we talk about the immune system coming up in a future unit. Now, another thing that can happen is that there's some characteristics that are influenced by numerous genes. Probably more than we even realize, okay? But this is called polygenic inheritance, and once you've got one characteristic that's resulting from additive effects of two or more genes. And a couple of examples are shown here. Eye color, there's a bunch of different eye colors that you can possibly have. Height, skin color, all of those are examples of polygenic inheritance in humans. So here we're going to talk about skin color, and then we'll do a problem about eye color in some of the problems that you'll work on. So our parent generation, there are basically 3 pairs of alleles that code for skin color. And so they range from somebody who's very, very light skin color and somebody with very, very dark skin color. The dark alleles are dominant over the light alleles, okay? So if you cross the person that is recessive for all 6 of those alleles, with a person who is dominant for all 6 of those alleles, what you'll find is that the F1 generation will produce offspring that are heterozygous for all 3 of those genes, and they'll be medium in skin color. There are a lot of different combinations, as you can see when you look around the population of people that you know. Okay, people from very, very light skin color to very, very dark skin color, and all different combinations in between. And it's based on how many copies of the dominant dark alleles that they have. Okay, and as you can see, the more copies you have, the more complex the Punnett square becomes, and the more complex the probabilities become as well. So we're not going to worry about doing those kind of crosses because they're very, very complicated in a big hurry. But it just shows you that that's why we have a lot of different combinations and so many different skin colors in the people that we know. When you plot that on a graph, okay, what you see is that in terms of the entire population, okay, there are only about 1/64th of the population that has the very lowest, the very lightest skin color and the very darkest skin color, and the vast majority have something in between, somewhere in between. And so just to show, it just has to do with how many copies of those alleles you inherit from your parents, not something that's under our control. And that is the end of the notes for this part.